Good morning, students. Our today's topic is biomacromolecules. In the organic compounds, mainly two type of fraction is there. One is acid soluble and other is acid insoluble fraction. The acid soluble fraction has the molecular weight ranging from 18 to around 800 Daltons approximately. So the acid soluble pool has the compounds which have the molecular weight ranging from 18 to 800 Dalton approximately. Whereas the acid insoluble fraction, which contains mainly four type of organic compounds like proteins, nucleic acids, polysaccharides and lipids. These classes of compounds, with the exception of lipids, they have the molecular weight of more than 10,000 Daltons and above. For this very reason, biomolecules, that is, chemical compounds found in living organisms, are of two types. One, those have the molecular weight less than 1000 Dalton and are referred as micromolecules, and other which are found in the acid insoluble fraction and are known as बायोमैक्रोमोलिक्यूल्स. अभी तक हमने क्या पढ़ा? दो तरह के फ्रैक्शन हो सकता है. एक तो एसिड इंसॉल्युबल जो हमारा फिल्टर के ऊपर रह गया था और जो फिल्टर हो गया था, नीचे फिल्ट्रेट आया था, दैट इज एसिड सॉल्युबल. जो अबव रह जाता है रिटेंटेट, वो होता है एसिड इनसॉल्युबल. जो एसिड इनसॉल्युबल है उसका मॉलिक्यूलर वेट 10,000 डाल्टन से अबव है, और जो कि एसिड सॉल्युबल है वो 18 to 800 डाल्टन तक उसका मॉलिक्यूलर वेट रहता है. द मॉलिक्यूल्स इन द इनसॉल्युबल फ्रैक्शन with the exception of lipids are polymeric substances, but the molecular weight of lipid is less. Lipid is macromolecule, considered as macromolecule, but still its weight is less than 800 Dalton. Why then why do lipids whose molecular weight do not exceed 800 Dalton come under acid insoluble fraction, that is the macromolecular fraction? So reason is lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. Lipids is the part of the cell membranes. So when we grind a tissue, we are disturbing the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. So when we crush the membranes, any living piece, we break the membranes that break the cell membrane into small pieces, and they form small vesicles. Small vesicles means small circular structures which are not water soluble, which are not soluble in water. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool, and hence in the macromolecular fraction, lipids are not strictly macromolecules. Protein, nucleic acids, amino acids, they are all macromolecules. Lipids are considered under macromolecules, but their molecular weight is less than 800 Dalton. The reason is because when we crush the membrane, it changes into small pieces and form vesicles which are insoluble in water, and they come in the insoluble fraction content. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Next is proteins. Proteins are polypeptides. Proteins are the chain of amino acids, that is, many amino acids combined together to form proteins. The different amino acids are joined by peptide bond, so it is known as polypeptide because many peptide bonds are present. So proteins are also known as polypeptide. Each protein is a polymer of amino acids. There are 21 type of amino acids, examples alanine, cysteine, proline, etc. A protein is a heteropolymer and not a homopolymer. Heteropolymer means it is made up of different type of amino acids. Hetero means different type of amino acids and not a homopolymer. When homopolymer means it is made up of same type of amino acids. So protein is a heteropolymer. In a homopolymer, only one type of monomer repeating n number of times. Certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The later, that is, the non-essential amino acids are those which our body can make, while we get essential amino acids through our diet or the food. So, amino acids can be of two types: essential amino acids and non-essential amino acids. Essential amino acids we take through the food or from our diet from outside, whereas non-essential amino acids, our body can make that amino acids. Protein carry out different functions in living organisms. Some help in transport nutrients across cell membrane, some fight infections, some are hormones, some are enzymes. So in the table 9.5, some proteins and their functions are given. Learn these protein, name of the proteins and their function. This is important for 1 mark. Collagen is the most abundant protein in animal world, and ribulose bisphosphate carboxylase oxygenase, in short known as Rubisco. Ribulose bisphosphate carboxylase oxygenase is the most abundant protein in the whole of the biosphere. So both the names are important. Most abundant in animal world, collagen. Most abundant protein in the whole biosphere is Rubisco. Learn these two. Next is polysaccharides. Polysaccharides are present in the acid insoluble fraction. Polysaccharides are long chains of sugar. They are threads containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharides, that is glucose. So cellulose is a polysaccharide which is formed from when many monomer units of glucose combine, cellulose is formed. So cellulose is a homopolymer. Homo means made up of same type of monomers. Starch is different of this, but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose. Learn the name. Inulin is a polymer of fructose. In a polysaccharide chain, the right end is called the reducing end and the left end is called the non-reducing end. It has branches as shown in this cartoon. In the figure 9.2, starch forms helical secondary structure. In fact, starch can hold iodine molecule in the helical portion. The starch iodine is blue in color. Cellulose does not contain complex helices and hence cannot hold iodine. Plant cells have made up of cellulose. Plant cell wall are made up of cellulose. Paper, cotton fiber is a cellulosic. There are more complex polysaccharides in nature. Chitin, which is present in the exoskeleton of arthropods, is a polysaccharide and it is homopolymer. Homopolymer means it is made up of same type of units. Nucleic acids are also present in acid insoluble fraction. They are polynucleotides That is, many nucleotides combine together to form polynucleotide. So together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. Nucleic acids, polysaccharides and polypeptides, they are the main components of macromolecules. Nucleic acids, in the nucleic acids, the building block is nucleotide. As already discussed, the nucleotide consists of three chemically distinct components. One is heterocyclic compound, that is nitrogenous base. The second is monosaccharide, that is sugar, and third is phosphoric acid or phosphate. So in a nucleotide, three components are present: sugar, phosphorus and nitrogenous base. The nitrogenous bases are adenine, guanine, uracil, cytosine and thymine. Adenine and guanine are known as substituted purines, while rest are substituted pyrimidines. A adenine and G guanine are substituted purines and other are substituted pyrimidines. That is, C substituted pyrimidines are cytosine, uracil and thymine. The skeletal heterocyclic ring is called a purine and pyrimidine respectively. The sugar found in polynucleotide is either ribose or deoxyribose. In RNA, ribose sugar is present, and in DNA, deoxyribose sugar is present. A nucleic acid containing deoxyribose is called deoxyribonucleic acid, while that which contains ribose is called ribonucleic acid. I hope this is clear. The structure of proteins we will study next time. Thank you.